Thank you so much. Yeah, it's really great to be here. Thank you for inviting both Stuart and myself. Unfortunately, we can't be there in person, but hopefully you'll have some idea about how Symbiotica is working, what we've done in the last 20 odd years, and what kind of projects we're working on. The format of this hour would be that I will give a short presentation of Symbiotica for about 20 minutes, talk a little bit about kind of the background that led to the establishment of Symbiotica. And then Stuart would give his perspective. And then with the time that's left, we'll be able to maybe have some conversation with the audience and maybe a conversation between Stuart and myself and some questions from anyone who's interested in learning more about what we're doing. So let me just share my screen. So we can start the formal presentation. Okay. Hopefully you get it now. Yes? Go. All right, so we titled the talk Contestiplinary Practices in Symbiotica because the way we see how we operate at Symbiotica is by engaging in a contestable mode of operation, creating contestable objects for cultural discussion first and foremost. And I would start with this little story. So in 2008, I was invited to give a talk in a Biofabricate meeting in New York. Biofabricate is a series of annual meetings of companies, startup companies and investors and other people who are interested in the future of basically biologically designed consumer products. It's something that I found quite interesting because, as I'll describe shortly, 25 years ago, I wrote my design thesis exactly about that. But I changed quite a lot of the way I was operating as I was learning more about biology, as I realized that the idea of working in and dealing with the questions that the new approaches to biology is raising is much more important and much more urgent than to try and develop consumer products. In that very meeting in 2018, the organizers set up a timeline of this new field of biofabrication and cellular agriculture. Cellular agriculture is a field where they're trying to basically produce animal products without the animals. And Israel is one of the places in the forefront of cellular agriculture, mainly through a number of startup companies that are attempting to grow meat in the lab, which is something that we've been doing also for quite a while, but for very different reasons. Anyway, in that meeting, the timeline was supposed to kind of describe the very field from its very beginning to where it is now and where it might progress. And I was very surprised to see a work that Yonat Zur and myself did in 2004 being put at the very beginning of that timeline as really the starting point for this whole new field and this whole new industry, if you like, that is now fledgling and starting to regain or gain traction. For me, it was weird because that very same work, the victimless leather, that made the rounds and was exhibited in MoMA in New York in 2008, was never intended to become a starting point for an industry, but rather was an object of a contestable object and an object for cultural discussion to see if this is really where we want to go. And obviously some people decided that that's the way. So I'll kind of try to give you some background as to why those kind of things might happen, why that work from 2004 was more than a decade in advance to any other kind of ways of thinking about using the technology, which is tissue engineering, for the purpose of making consumer products. Now, the idea of biology moving from a merely descriptive discipline to a prescriptive discipline, the idea that biology should become an engineering pursuit is not that new. This guy over here, Jacques Laub, who was a German-American scientist, already in the early 20th century was talking about his desire to see engineering moving towards, biology moving towards an engineering pursuit. And this idea of biology as engineering and life becoming a raw material for human exploitation and for the extraction of value from biological systems is something that we've seen very slowly progressing throughout the 20th century, and obviously now exploding at the first decades of the 21st century. And for Yonat Zur and myself, it's kind of an interesting thing because often what we see with this attempt to make biology into a technology, life into a raw material, that it's becoming like a fetish. So the fetish of technological approaches to life often overshadows the context in which life operates. Yonat and myself call it Neolifism, this form of fetishization of life. It seems that the biological milieu is transformed into an abstract technological instrument of control, where life is just another raw material to be engineered. Decontextualized life has been configured, mixed, remixed, reappropriated, and instrumentalized to such an extent that often technologically imagined potential of life stand for what life is. And we feel that this is an issue that needs to be explored. A few years ago, in 2017, the World Economic Forum came up with a document dealing with how to deal with this new industrial revolution, what I refer to as the fourth industrial revolution. And even though I have some issues with the way the World Economic Forum operate and the motivations and agendas, I think that they hit a really important point here where they say that we face the task of understanding and governing 21st century technologies, and I would add 21st century knowledge as well, with 20th century mindset. And this is important. The mindset bit, I think, is something that we really need to explore further. And obviously trying to operate from within 19th century institutions. So from their perspective, institutional change is critical, but I would say also a cultural change is even more critical to try and make sense of those new shifts in the way we think about the world around us, and in my context, the way we think about the life sciences and life in general, and how our relationship to the concept of life is lagging behind. So as a culture, as a society, our idea of life is lagging behind what scientists tell us about life and what we then decide to do with life through the technological application of that knowledge. And it's something that we have to acknowledge, and we're in kind of a perfect storm at the moment, because humans are suffering from a psychopathology of control. We already recognize that human-made technology that was developed from the bottom up, is becoming so complex that we are losing any way of controlling it. We already relinquished our control over so many human-made systems, be it our economy, be it machine learning and different types of algorithms, that even the people who programmed those systems have no idea how they actually operate. And obviously we're now even giving technology the ability to make life and death decisions, think about kind of the ethics around driverless cars and killer drones. So there are already technological systems that are making their own choices in regard to even issues of life and death. At the very same time, we are now in the midst of trying to assert our control over systems that existed outside of human intervention for millennia, and that's living biological systems. So complex systems that we are now trying to figure out how we can make them work for us, as some of the synthetic biologists would say. They want to see life becomes a well-oiled machine. They want to see, they refer to evolution as a tyranny, and they want to see how they can control life and make life into this new mode of production, and we need to address it. So there's an interesting kind of a reversal in roles if you think about kind of the first industrial revolution, where one of its main outcomes was to transfer labor from sentient biological agents, be it workers, slaves, or farm animals, into non-sentient machines. Yeah, the steam engine, the milling machines, all of those systems that were developed in order to make labor and work easier, consistent, and obviously with machines that don't complain, don't feel pain, and don't sense the world around them as much. But what we see now with this so-called fourth industrial revolution is that it promises to bring sentiency to machines through artificial intelligence, and at the very same time remove the sentiency from biological entities through biotechnology and synthetic biology in the name of increasing productivity and efficiency. So again, think about this whole field of cellular agriculture, that one of the main premises of that is that we will be able to basically produce animal products without the animals. And one of the main arguments of this new field is that those cells in culture that are growing meat, for example, or any other product, are not suffering pain or anything like that. They are not sentient, and therefore we can exploit them as much as we want. Now, as an artist, I'm really interested in the question it raises, because it reveals that this kind of reversal in roles changes and raises issues that we really need as a society, as a culture, to come to terms with. This is the mindset that I referred to before. So we need to really deal with the implications of what it means to the human and non-human relationship. Non-human can be, doesn't necessarily have to be a living non-human. How do we deal with issues of sentiency, life, labor, and exploitation? And we have to remember that with all of the rhetoric of the new biological kind of industry, where they talk about the fact that it's basically like nature does it, and that was also part of the main premise that 25 years ago I was trying to kind of explore through my design thesis. The idea of the fact that it's natural is not correct, because all of those are attempts, like Jonathan Safran Foer said, are basically considering nature itself as an obstacle to be overcome. So we need to maintain that in mind. So my original 1996 design thesis was basically a speculative design project where I was speculating a future in which designers would be called upon to design living biotechnological products, and how kind of designers would start to think about what it means to design living things. You don't really need to read what that is, but this is kind of a letter I sent in 1996 to people around the world to get their advice. I was kind of trying to explore something that I felt that there was very little knowledge about, and I was trying to get as much responses as possible. But what I found quite interesting is looking back at the questions I raised then, was that I already identified an issue that seems to be addressed, that needs to be addressed, and that's the ethical issues and the way we relate to life. As I was doing my research, I was looking at many different types of biological technologies that were coming to age at the time in 1996, things like genetic engineering, different types of plant tissue culture, working with bacterial mats. But one technology in particular grabbed my attention, and that's the technology of tissue engineering, or as it's known now as regenerative medicine, because that really generated a major shift in the way we think about bodies. As Tomer mentioned, I spent a year in Harvard Medical School learning from one of the top scientists and actually the founder of the field of tissue engineering. And those slides are actually from a presentation that he gave in the year 2000 in the third International Tissue Engineering meeting. And he was very proud to show how the field really changed the way we think about bodies. So the image to your left is how we imagine the body is going to be repaired in 1989. What we see here is basically attempts to replace missing or failing body parts with dry, hard, mechanical or electrical engineering devices that bit by bit would replace the body into kind of almost like a cybernetic robotic entity. Think about the Tin Man from The Wizard of Oz, something like that. And then exactly 10 years later, in 1999, as tissue engineering was progressing, the body became its own regenerative site. The body became its own technology to repair itself. The idea was that you would start to find ways to harness those latent regenerative powers of the body in order to be able to grow body spare parts. The original idea of tissue engineering was very simple. It was basically developing different types of biodegradable polymers, which you would then shape in the desired shape of the organ you're trying to replace. You would then seed it with the cells that you are of the organ and grow them in vitro, grow them basically in a petri dish outside of the body for a while, and then implant it into the body, and supposedly you'll have a new body part. Now, 20 odd years later, there's still very, very little in progress in that field, in the sense of there's very few clinically available organs that are grown in such a way. And the organs that are available are extremely simple. So things like bladders or maybe some bone replacements, a cartridge, but the complex organs are still eluding our ability to be able to regenerate them this way. But in my design thesis and later on with the project that I developed together with Yonat Zur, we were never interested in this idea of putting things back into the body. We said if the premise was to be able to grow complex organs outside of the body that would function and grow in 3 dimensions, why not to grow products that would exist independently from the body and would have the functions that you're looking at? And that's when we came up with the idea of the semi-living entities and semi-living products. But we felt that the questions that those types of issues raise are much more important than to try to find anything that is so-called useful, because we need to come to terms with what does it mean to have those entities which are basically parts of complex bodies that are living and existing outside of the original body with the support of the technological apparatus, with this idea of the surrogate technological body, and how we kind of would think about sharing the world with those new types of entities. So we were very lucky. I came up with this idea to a scientist, which was actually Stuart's postdoc supervisor, Professor Miranda Grounds, with the idea of looking at the possibility of using tissue engineering as a medium for artistic expression, and look at the possibility and the ways in which one might be able to grow 3-dimensional tissue sculptures. And instead of throwing me out of the room, she was very curious, and she invited me to work in her lab and introduced me to many other scientists. And shortly after, I met Stuart, and we started to work together on different projects. And the modus operandi that we decided to go with, and that was a lot to do with kind of an ethical question as well, about the responsibility that one has towards a living system that one is engaged in engineering and manipulating. So we wanted to do it ourselves. We didn't want to commission scientists to do the work for us. We wanted to learn from the scientists and then develop our techniques and develop ways in which we can grow our own tissue engineered sculptures. That proved to be quite successful. Tissue engineering is not rocket science. It's very much a craft, and it's very much based on techniques that we often talk about. It's like a combination between gardening and cooking and looking after a living thing. So this idea of care is extremely important in the process. So we started to work in the lab. We had a series of exhibitions, and especially after our 1998 debut exhibition at the Perth Institute of Contemporary Art, many other artists approached us and asked us, how did you manage to get access to the labs? How did you manage to work with scientists? And we said it was very easy. We just went there and asked them, but then we decided to develop a place where artists can come. There was something else which was really important for us. When we came to the University of Western Australia and to Miranda with those ideas, we always felt as if we were asking for favors. The power structure was very obvious, that we were only guests, that we didn't really have, or we could be kicked out of the lab in any time. And when those other artists approached us and we decided to figure out a more institutionalized way to support those types of practices, we were able to apply for funding and get some funding from the West Australian Lottery Commission to build a dedicated space, which we called SymbioticA. And we modeled it after basically a biological research lab. It was based in, at the time, the Department of Anatomy and Human Biology, which is now the School of Human Sciences. We positioned ourselves as yet another lab in this whole complex of the school. So among all of the other labs, there was also a biological lab that dealt with artistic research into biology. It was really successful in the sense that, you know, we're still here, we still are doing work, and we are treated as equals. So we were able to negotiate access to ourselves and all of the researchers that then came and started to work with us, which was equal. So it wasn't anymore about this power relationship. It was really about the fact that this type of artistic research, it took us quite a lot of time to convince people, but was then seen to be as valid as any other research activity that took place in our school. And here you can see some images of our lab. We, as far as we know, we were the first level 2 biological lab that was dedicated specifically for artistic research, and now there's a few others around the world, but that was kind of very early in the game. And we set it up as an artistic research laboratory that is dedicated to the learning, critique, and hands-on engagement with the life sciences. We strive to articulate and provide a new cultural language to those shifting relationship and perceptions of life with our contestable artistic interventions that can come in many forms. So kind of just to recap, we explore those philosophical ideas around those shifting perceptions of life. We spend a lot of time trying to identify and develop new materials and subjects for artistic manipulation. So we expanded our interests and the interest of the lab from working with tissue engineering to a wide range of biological interventions with life or technological interventions with life. So we see ourselves now as a research lab that deals with life from the molecular to the ecological, but all of the people who are coming to do research with us are committed to learn biological techniques and spend time in the lab to have this direct experiential engagement with the living systems that they are dealing with, both as the subject and the object of their manipulation. You can imagine that as biology becomes more and more of an engineering pursuit, there's a couple of things that happen. One of them is the de-skilling. So there's better ways to do things. You don't need to have a PhD in biology now to do genetic engineering. There's quite a few standardized ways to do it. So the standardization that comes from an engineering pursuit actually help us as artists to develop different other ways to intervene and subvert it. And also, obviously, it opens up a huge kind of palette of artistic possibilities where the manipulation of life is both, as I mentioned, the subject and the object of our artistic manipulations. Another thing we do at SymbioticA is research strategies and implications of presenting living art in different contexts. You can imagine that the idea of taking manipulated life forms outside of the containment of the lab and putting them within a cultural context generates quite a lot of very interesting reactions and resistance. Museums and galleries, which is kind of the natural place where we're showing our work, are very much 19th century institutions. The mindsets, I hope, in many cases didn't even reach the 20th century. So try to bring those types of new ways of thinking about life and art can generate a lot of tension, and we often work with those institutions to try to develop strategies where this kind of work can actually be displayed in those cultural contexts. And sometimes we also develop our own technologies and protocols as an artistic toolkit, because often the considerations, the concerns of the artists are very, very different than that of the scientists. And the tools that are available are obviously geared towards scientific research and not so much for artistic research. So we do develop tools for artistic research and display of living biological systems within those contexts. And here you can see a very partial kind of taxonomy of the areas of engagement that we already developed at SymbioticA since our establishment in the year 2000. We had more than 120 residents and researchers, and more than 20, actually, I think by now it would be 30 or 40 postgraduate students that are undertaking many different projects. And I won't have time to go over the projects they're doing. Stuart will mention a couple of projects that he was working on. But just to give you an idea, it's ranging from molecular biology to neuroscience to anthropology and science and technology studies to ecological systems and so forth. So, and the way we also set up SymbioticA is, as I mentioned, we set ourselves up as a research lab, but we also offer academic courses, and we also produce exhibitions, residencies, offer research residencies, and organizing symposia, seminars, and workshops.
So this show was set to commemorate the 20th anniversary of the first public appearance this very curious creature, the mouse that had a human ear growing from its back. And the idea was to look at the last 20 years and see what kind of cultural impact this image had, both within kind of artistic and cultural context, as well as popular media and popular culture, and as well as, to some extent, the scientific discourse around it. As an artist, I was very intrigued by this image, and I must admit that my own practice, my own artistic practice, owes a lot to me being exposed to this image 20 years ago. What we're trying to do here is to tell a story of the different ways in which one can see this ear on the back of the mouse. You know, to some extent it's for everyone, because when the image on the back of the mouse, and actually we have the original BBC documentary from 1995 when it was aired for the first time, and that was kind of a popular science show talking about the future of repairing bodies. And you know, we all are concerned about how we're going to repair our bodies. So this is one aspect. So in a sense, I didn't try to aim it to a specific group of people. The idea was to look at the way artists of many different areas of specialities and expertise responded, and either spontaneously to the ear on the mouse or by us approaching them and see what they would come out with. You can't avoid the fact that it is a very striking image, and for me as an artist it really represents the surrealist dream comes alive. And what was interesting from my perspective, the reason why I got interested in that as well, is that this was developed by scientists, presumably for a different purpose, but with a very, I suppose, strong inclination into trying to capture the public imagination.
So that was an example of an exhibition that I co-curated in 2015. And we, as I mentioned, we are organizing quite a few exhibitions. In 2018, we had a fairly major festival here in Perth commemorating the bicentenary of the publication of Frankenstein. So we do try to find kind of thematic areas to curate shows and organize conferences and exhibitions around those topics. As I mentioned, we run an academic program, so we are, as far as I know, the only place in the world that actually offers a Masters of Biological Art that we refer to as the more useful and the real MBA as a science degree, but it's basically the people we cater for can come from either a scientific background or an artistic background, and they spend 2 years doing research into biological arts. And I'll stop here, and I'll now pass the presentation to Stuart, where he can talk about his experiences. And as I mentioned, we would then talk a bit more about how we're doing things. And if we have time, I might be able to go back to a couple of the projects that I've been doing in the last 20 odd years. Thank you.
Thanks, Aaron. Just let me interrupt 1 second. I just wanted to say that I'm not sure that in this session we'll have time, but the audience can ask questions through the chat box right next to the on the YouTube page. So feel free to write and ask questions, and if we'll have time, I will read them towards the end of the meeting.
Okay, so thank you very much for the opportunity to speak, and thanks to Aaron for originally getting me involved in this adventure over the last 20 years now, I believe. I'm just going to share my screen so I can start my talk. Can you see that? Yep, good, okay. So my name is Stuart Hodgetts, and I'm the director of the Spinal Cord Repair Laboratory at the University of Western Australia and also the Perrin Institute. Just in the same way that Aaron started, I think it's important for you to have an understanding about my background. So I did my PhD way back in the 90s at Essex University, studying autoimmunity. And my first postdoctoral position was also at Essex University, where I did plant molecular biology. So I actually started to sort of look at plants in the molecular level. I then did another postdoc in Oklahoma City at the Medical Research Foundation there, looking at immunobiology and cancer. And then because I'd left my family for a long, long time, I decided to come back to Western Australia. And in 1998, I managed to secure a position at the University of Western Australia in the then Department of Anatomy and Human Biology, which is now Anatomy, Physiology and Human Biology within the School of Human Sciences. And in 2015, I also became part of this Perrin Institute for Neurological and Translational Science. And you can see down here, what a lovely building here with really large neurons on it. So that's where I spend half of my time. My research interests are really strategies around trying to repair the spinal cord following injury. And I guess the bread and butter of my work is really in this sort of not fully comprehensive list, but certainly stem cell transplantation back into the spinal cord is one of my main therapies. And we're using more and more now gene therapies to try and induce plasticity and promote regrowth and regeneration, both in the brain to help go down towards the spinal cord and in the spinal cord itself. And we're combining these treatments along with others such as tissue engineering, as well as non-invasive therapies like magnetic stimulation and infrared light, to try and promote this repair of the injured spinal cord and really try and get translational therapies. As Aaron mentioned, this is now called regenerative medicine. A very recent embarkment that we've gone on is to try and use, this is important because Aaron was talking about how people tried to use technology to replace parts of the human body that were defective, and then the switches to sort of come back and try and promote regenerative biology. We're now actually able to, in the lab, to try and take viruses and stick these viruses back in the body, and they are the ones that re-engineer a particular tissue into another type of tissue in order to do that work for us, rather than relying on stem cell transplantation. So that's a really interesting new focus within the lab. But essentially it's strategies to try and teach spinal cord repair. And my other role is also chair of the Animal Users Group at the University of Western Australia. So I interact with other scientists that use animals for their research. And of course, it means I have to liaise with veterinarian staff and the ethics committees. One of my more important roles I consider, and certainly one I'm very, very proud of, is to be scientific consultant to Symbiotica. And as Aaron said, this is a long time association. And in terms of my role with Symbiotica, I'm basically a scientific advisor, not only to Symbiotica, but also to the residents, and a mentor to the residents. I do a lot of assessment of residents' proposals in terms of reading their proposal and trying to find out whether or not we can actually do the work. And then when they come, actually being able to provide some sort of network both within University of Western Australia and outside of UWA. So if I'm not the right person to help, then I can find somebody who's actually willing to dedicate some time and potentially money to help the residents. But very often I do try and directly assist with the projects myself, because I find the interaction with the residents extremely stimulating. And so I try and help wherever I possibly can. But I also am involved quite closely with just checking on ethics applications and biosafety procedures to do with these proposals. Another important part of my collaboration with Aaron was also being part of tissue engineering workshops in Europe and Australia. So multivariant sort of approach there to helping with Symbiotica. And I've also been involved with projects myself, both helping people behind the scenes, but also being part of the exhibition. Tissue Culture and Art Project is one. Pigwings, which is an extremely important piece of work from Symbiotica. And later on, I actually managed to go out to Norway myself as a team member for Lifeboat. You can see here I'm letting my hair down on the water there in Norway. This is an extremely challenging project for us all, and a very rewarding project as far as I'm concerned in terms of the success. So not only do I interact with Symbiotica, but I also interact with other artists. Guy Ben-Ari, who's a long-standing relationship with Symbiotica as well. I've been involved with some art projects that he's been producing. Impotentia is one of them. And I've just given a link to the projects at the bottom right-hand corner if you're interested. And one that's gained a lot of attention recently is Self, where we've basically been able to perform a surrogate performance using cells that were taken from guys' skin and reprogrammed into neurons. And you can see we created this device, which is essentially a mobile lab, which takes analog inputs from musicians that play music to this neural network that we grow on these chips. And we can listen to the real-time output as the essentially the cells inside the chip jam with the musician. So this is an important development in terms of the science that we were able to provide for people like Guy and Nathan and Darren. And more recently, a new project that Guy and myself and Nathan Thompson are embarking upon is a process called revivification, where we're taking cells, skin cells from Alvin Lucier, who's an extremely well-known landmark performer. We now have his cells growing, you can see in the middle here, and we are going to be turning them into neurons. So this is an extension of a process like self. So these are really important landmark projects. And this is one of the main reasons why I remain involved with people like SymbioticA and other artists, is that they're extremely stimulating scientifically as well as artistically. And of course, in terms of the scientific side of this collaboration, a lot of scientists really are not forced to ask themselves very basic questions until they interact with these residents. What is artistic research? How does it differ to scientific research? These things are extremely close in some respects, but also extremely different. And what are the outcomes of the artistic research? Are they the same as the scientific ones? And should it follow the same principles? So a lot of scientists are not really familiar with artistic research. So I consider myself very fortunate and very blessed that I now have a much greater understanding of artistic research. But most scientists, unless they're approached and do work for the first time with people like SymbioticA and other artists, really don't know about art as an artistic medium necessarily. And of course, Bioart really is a marriage of biological and biological medium and a process of ethics with art, governing welfare and the humane use of biological mediums, whether it be human or animal, as well as biosafety. And in my capacity with SymbioticA, I basically provide guidance towards hopefully a successful marriage of these two by assessing the feasibility of the proposed work scientifically, logistically, whether we have the infrastructure, the networks, as well as dealing with ethics applications. This process then often becomes part of the artistic residency itself, and arguably for some residents, they've decided to take this on as a medium in of itself. So it does in effect become training with the medium for the artist or the resident to perform bioart within the university. Of course, my role is not to judge. I'm there to provide some sort of scientific merit of the artistic proposal, to decide whether or not the use of that biological medium is warranted is really something that's not necessarily my avenue, but I do provide an avenue for residents to try and achieve their aims wherever possible. And of course, it's really important to understand that it's not just performing the work with a scientist. There are ethics and there are biosafety committees involved as well. And of course, the impact of their work can really influence whether or not these committees need to make a decision of whether or not the artistic proposal itself has merit as opposed to scientific proposals. So they may actually have to make decisions about whether or not they consider this use of this material is actually warranted. So some of the problems that we encounter with residents revolve around this. And at the basic level, very simply, can the work be done here? So I try to provide resources as well as give infrastructure. I try to provide information about who can help within the university or outside. And again, look at ethics, biosafety applications, completion of those, and whether or not the outcomes of the research project are going to be achievable. But of course, at a much higher level, there are requests to ethics and biosafety committees to allow that work to proceed, and whether or not those people are able to then go ahead and do their work on time and complete their projects. The use of spare animal tissues is obviously a way around this rather than using live tissues, but again, can be problematical. And in terms of using biological material for artistic research, the use of animal tissue in particular is advantageous for the ability for scientists certainly to provide proof of principle in vitro or as a relevant alternative model to in vivo studies. So can we do something using animal tissue outside of the animal that's relevant? And for scientists, the responsibility is for them to publish the data that they use from this work. Because ethics requires us to consider the animal's inability to consent for itself to be used for these research purposes. So one of the big problems that I'm certainly aware of now is what is the impact on those ethics requirements in terms of the decision as to whether or not the use for art is actually outweighs the use for science. And again, some important questions about this is really who gets to decide these. Should it just be the responsibility of the ethics committee members? Should others be involved? For non-scientific use, of course, we have to justify things. It has to be outcomes based. Is it a hypothesis-driven design? What's going to be the waste of the animals? And so the inability to provide satisfactory outcomes or answers to these questions when we talk to other scientists about helping artists, it's often perceived by some scientists as not necessarily appropriate or a good enough reason to use this sort of material. And so scientists can often, and certainly this happened with Symbiotica in the early days, was to scrutinize the worthiness of these types of projects. And of course, artists are rightly scrutinized by public and scientists themselves. Certainly ethics and biosafety committees would be part of that. But also scientists who help artists have often been scrutinized by fellow scientists in terms of being guilty by association for wasting their time with artists who want to do this type of work that they consider is not necessarily appropriate. And of course, this has potential impact on ethical status or well-being. So again, is this work justified is one of the biggest problems. But occasionally, members of the two disciplines find common ground. Artists employing research techniques, I very often have an argument with Oron about this. I consider them disciples of science to some degree, and certainly budding or kitchen scientists in their own right, their work certainly does have scientific merit, and so it is worthy. And so the potential impact on the ethics and biosafety committees can very often be outweighed by these types of productivity. And certainly scientists can generate currency from their own involvement in work with artists. I, for myself, have been able to generate publications and successfully with Unat Zur obtained money for grants. And also more recently, one of the big benefits for me is certainly that I've been able to take some of the ideas from these artist residents and actually incorporate them into education at an undergraduate and sometimes postgraduate level. And the text that I've given here just gives you some examples of that. So certainly there is a very important role to play for these types of residencies and the work that's being done in terms of generating currency for scientists to use as well. So it certainly is an extremely productive collaboration in that respect. And I'd like to sort of finish off by using some text that Unat Zur and I presented at a meeting to animal ethics committees many years ago now, but I think it's still very relevant today, in that this artists have this essential role in exploring these new terrains by continuing to question and provoke, certainly by the types of medium that they use, contestable scenarios. And certainly in terms of regards of ethics committees, methodologies, which are geared predominantly for scientific research, it becomes a great challenge when we apply this to the art, and certainly biological art. So the weighting projects benefits versus costs becomes more of a philosophical issue when applied to the artistic or cultural activity. And new issues are raised, such as the role of the committees as censors, protection of the audience sensitivities, perception of artistic research versus scientific research, and allowing this sort of playful research to be recognized as a serious research tool in its own right. And certainly there's tension between the artistic research and the cost benefit analysis in terms of the universities and their ethics committees. And there's an increasing use of living matter as technology and treating life as raw material, as Oron said, that can be manipulated and engineered. And the human relationship to life is certainly increasingly confronted by our ability to intervene at all levels of this life process. And just as engineers are entering the field of life sciences to offer engineering solutions and technological solutions and utilitarian approaches, so should artists who offer non-utilitarian artifacts and gestures be able to also contribute to this process to problematize and provoke and subvert these dominant understandings and uses. And I'll leave it there. Thanks very much.
Thank you, Stuart, and thank you, Ron, for this fantastic and mind-blowing set of ideas and collaborations. We only have a few more minutes, and there are questions both. I see one at the on the YouTube channel and a few from here. Let me just repeat the question that is being asked on the YouTube channel. Someone is asking, is an artist at the lab a scientist? How do you view yourself, I guess, Ron?
Shall I start? So yeah, Stuart alluded to the fact that we often have arguments about it. I don't believe that, you know, I've been spending 25 years in labs. I've been working with scientific tools and techniques, but I would never claim that I'm a scientist. The furthest I'm willing to go is that I'm a researcher. I'm producing new knowledge, but my methodology is very, very different than Stuart's, for example. So we can work side by side in the sterile hood, do something that would look almost identical, but because of my motivations and my methodology and what I would like the outcome to be, I would never claim that I'm a scientist. And actually, we won't have time to go into it, but Jonathan himself wrote quite a lot recently, in particular, about kind of the social contracts that we have when we present ourselves as either artist or scientist. The context in which we present ourselves really change the reading of what we're saying. And it's extremely important, especially in a time now, like now, of post-truth and mistrust in everything, and the fact that so many people live in fictional kind of realities and speculative kind of way of being in the world, that we do have some grounding and understanding that if I tell you something as an artist, you should read it in a very different way than if Stuart would claim something as a scientist. Now, at SymbioticA, we make sure that our claims are actually grounded. That's our choice. But by definition, I would say that artists are never obliged to tell you the truth, in a sense. Artists are being celebrated for making things look like something they're not. And if a scientist would try to do it, they'll be punished.
Yeah, I think, and I totally understand that, and I appreciate your side of the argument. I would just counter with one thing, and this is something that a colleague of mine, Alan Harvey, has said recently, was that in the old days, there was never this differentiation between art and science. They were always considered essentially the same thing. And at some point they diverged and gave rise to these two labels. And like most well-trained scientists, we like to give labels to certain things. And so if we see somebody who doesn't have a scientific qualification performing a scientific methodology, whether they like it or not, we're going to call them a scientist. Whether it's a budding scientist at an undergraduate level who's just managed to just screw up their tissue culture project for the first time and waste thousands of dollars worth of equipment and media and all these types of things, or whether they're you know, an expert like myself who still manages to screw up every now and again. That methodology, rightly or wrongly, I think has a scientific label that's attached to it. And sometimes scientists would like to be fondly considered as artists too. So I certainly think it can go both ways.
That's even more dangerous, actually. you know, the problem I have is that if this is really what you're trying to do, if you try to make artists into scientists and scientists into artists, you would get really bad art and really bad science. You know, I think first of all, we have to respect the fact that, you know, I have years of training as an artist, Stuart has years of training as a scientist. It changes the way we see the world. It's change our intention when we engage with some of those questions. We can work together, But I believe that, and that's something that I constantly talk about at SymbioticA, and that's the need for maintaining an integrity of disciplines when you work as a collaborative kind of team. Otherwise, it can get really messy, and the outcomes are often not that interesting because you have this slippage that more often than not is not really helpful. But this is kind of one of the idea of the kind of contestiplinary approach that we have here, because we constantly contest our own roles and in those kind of relationships, as well as obviously we're contesting some of the knowledge that is being generated, as well as some of the ways in which we choose to present this knowledge.
Fantastic. We are almost running out of time, but I think maybe I can ask a question which might not have a short answer, but let me try and see until while we're waiting for the next set of speakers on this channel. So we all know that innovation is central to rapid social and economical development, and the problem is that often economical pressure is influencing fundamental research. So I wonder if you could comment on the benefit of fundamental research and your role as an artist, or both of your roles, using art to convey this message.
All right, let me start again, stories. So there's one of the things is obviously with what we've seen happening to the world in the last year, exactly a year from today, the first COVID case was discovered. Now, I would claim that if we would have invested more in fundamental research and understand viruses, not because we have a direct and immediate danger from them, but because we want to know more about how kind of life operates, including viruses, we would be in a much better economic position than if we would work according to this paradigm of innovation that it's tend to work towards short-term knowable kind of outcomes. Yeah, so with all of the due respect that one might have towards innovation actually, in my perspective, closing down a lot of new ways of seeing the world and engaging with the world. And actually, this drive for innovation is actually opening up a new dark age, because it really put blinkers on the way we see what we can do. And if everything is being done through the lenses of extractive and exploitative notion of trying to generate short-term kind of value out of systems, we'll never be able to break through it. And I think that the other issue with innovation, it's obviously, especially in the last few decades, is that it adapts the Silicon Valley ethos of fake it till you make it. So people have to actually make claims about things which are not really either scientifically true or achievable, and the whole idea is that you get the money and then you try to figure it out, but you claim that you already done it. And what we see now, especially around the field of cellular agriculture and biofabrication, that because of the early stages of this industry, the narratives that are being given to us are basically narratives of startup companies trying to attract investors, while the general public is basically a collateral damage. But the problem of that, it also prevents us from thinking about other solutions which are not driven by innovation, but driven by behavioral change. And if there's anything COVID taught us, is that we can change our behavior if we need to, while the idea of innovation is to continue with business as usual, because solutions would always allow us to continue exploiting the world in ways that in many cases are fantastical, in the sense of they're not, they have no grounding in scientific knowledge or in the way we can think about the world in general.
I would certainly echo that, and very, very quickly, just to try and give it a new label. We've all heard about fake news. The claim to try and make things innovative in order to secure funding is actually what I would consider fake promise, and really needs to be removed like a cancer.
Right. Okay. Thank you so much, Ron and Stuart, for being with us and for the fantastic talk and the project. And we'll now move on to the next session. Thank you.